No, I mean, I don't think so. Do I? Do we? I don't think we have. I think we had. Yeah, we don't have. Yeah. No.
People might not appreciate that, but I appreciate you saying that.
Okay, good morning, everybody. We're going to get started very soon, and we are going to start our day with a fantastic presentation by a remarkable pioneer and leader in the field of brain mapping. So today we have our keynote speaker, Dr. Paul Thompson. Paul and I go back many, many years. I would say probably almost 2 decades, but so he created this ENIGMA program consortium, which is truly fantastic, and across 45 countries, and they have done remarkable work. And Paul is also highly respected professor at University of Southern California, where I am also graduated from. So with that, it's an honor to pass the podium to Professor Paul Thompson.
Appreciate it very much. Thank you. All right, good morning, everyone. Really appreciate you attending this early Sunday morning session, so we'll try and make it fun for everybody. So I'm going to spend about 30 minutes or so taking you on a guided tour of some brain mapping projects that are going on throughout the world and have been going on for the last 15 years or so to understand some of the major diseases of the human brain. So all of you know this. There are many techniques to map the living brain from its structure to its function to its molecular content. And there's been a huge push to collect population data from all these different devices to understand what the course of development and aging are, what the metrics are that we can use to identify whether a patient is getting better, and also to understand how the brain measures relate to pathology, to blood markers, to other behavioral data, and even genetic data, even the variations in our genetic code. So the ENIGMA Consortium was founded in 2009, and it's published the largest neuroimaging studies of now 15 brain disorders. And the goal of the consortium is to understand factors that help or harm the brain, basically by analyzing brain scans and genetic data from a little over 100,000 people now. Now, no one site can collect that amount of data, so 500 medical centers that are scanning their patients are contributing from 45 countries, and there are over 2,000 scientists analyzing this data, not all in one group, but they're in about 50 different working groups, and this has led to the largest neuroimaging studies of epilepsy, of Parkinson's, of ataxia, of traumatic brain injury. And then in psychiatry, the largest neuroimaging studies of bipolar, major depression, schizophrenia, PTSD, substance use. And then in the developmental area, OCD, ADHD, autism spectrum disorder, and some neurogenetic syndromes such as 22q11 deletion syndrome, and also brain mapping of infectious diseases, which I'll talk about a little. So ENIGMA's analyses of 100,000 brain scans have had 2 main goals. So one is, how do the major disorders of the brain affect the brain, at least as far as we can see with MRI, DTI, resting state fMRI, and also with EEG and MEG and spectroscopy. Another big challenge, and this was how ENIGMA began, was to discover how does genetic variation affect the brain. I mean, we all have differences in our DNA. Some of those differences affect our risk for brain disease or our treatment response. So we've been looking for genetic markers and environmental factors. I mean, there's also effects outside the genome that matter for brain development, aging, disease risk, and treatment response. Now, why would you look at 100,000 brain scans? I mean, not every clinical question requires that amount of data. One of them was to develop the statistical power to answer new kinds of questions. So if I was to say, is there any variation in your DNA that impacts the maps that we collect in brain scanners. I mean, that obviously is a difficult question that would need high statistical power. It's been impossible to solve before. It is very efficient, so you can use computers across the world and expertise across the world, collective intelligence, so there are other benefits too. So just to sort of give you a roadmap of what we'll talk about what is ENIGMA, how do the neuroscientists in different countries work together. We'll talk about worldwide genetic analysis of brain images, how to link neuroimaging with genetics. And I'll show you some highlights from global studies of brain diseases, along with the challenges of pulling data internationally in some new directions with AI and studying treatment. So this is where the data is. So the data is not at USC. In fact, it isn't at any one university. So all these neuroimaging centers in 45 countries are scanning their patients. The colors are the different diseases. So like we said, there's patients with neurological, developmental, psychiatric conditions. The color and the key is what is the disorder that each center mainly studies. I mean, many academic hospitals study many disorders, but the cohorts are color-coded there. And there's a lot of reviews, some in Science, Neuron, other journals, about how this came about and how the logistics work of putting this data together. But it's not centralized. It's not centralized data. And it leads to a lot of papers. So last year, over 70 papers from ENIGMA were published, including a big special issue of the Human Brain Mapping Journal that talk about how you would bring scientists together to merge their brain scans. So here are the working groups. So in ENIGMA, the diseases that are studied are shown here. In the middle, you see neurology. So there's brain injury, ataxia, FTD, Parkinson's. There are other groups that are natural allies. And so on the upper left, you'll see bipolar, schizophrenia, MDD. But a lot of the studies of those conditions would be illuminated if you also were studying other conditions, people with early onset psychosis, maybe people with suicidal thoughts and behavior, and then over on the right, people with panic disorder, social anxiety. Now, you'll notice that a lot of these conditions, there are not big brain imaging studies. Some there are. I mean, certainly Parkinson's, there's big brain imaging studies. But for many of them, this was the first time to come together and do a really high-powered brain scanning study to understand what's going on in the disease. And I don't want to oversimplify. I just want to say some of these conditions are very heterogeneous. So a brain injury could happen in the military, in sports. It could happen to a young child, sadly through intimate partner violence. So there are many sources of data on these different brain conditions that we have to respect, that they might fundamentally be different. Now, there are a lot of techniques you can use as well. So I talked about the clinical disorders. There's techniques. So on the imaging side, there's fMRI, EEG, I mean, all these methods. On the genetic and omics side, there's GWAS, there's epigenetics. So when these types of data are collected from the patient groups, there are special support groups that help analyze data of these different kinds. And these are really the whole spectrum of groups in ENIGMA today. In the middle, you see neurology, there's brain trauma, ataxia, stroke. Over there on the bottom left, there's psychiatry, there's PTSD, ADHD, many other conditions. And all of these groups are talking to each other, consulting with each other about what they're finding and using common methods. And here are some of the folks that lead ENIGMA's efforts. So what do you see in the brain map? So these 9 disorders, these are areas of the brain that are different in patients with these different disorders. And the key is MDD is major depression, BD is bipolar disorder, schizophrenia, alcohol use. And the map that's being shown here is a map of gray matter deficits. So this is not a functional map, it's just simply the structure of the brain. Are there any systematic differences in these disorders? And many of these are the largest studies to date of, for example, obsessive compulsive disorder, autism spectrum disorder, epilepsy and schizophrenia. And you can see how very different the signatures in the brain are for the different conditions. Now, not everyone's the same, and we'll go into some depth about the variation among patients with these conditions. Okay, so, and there's even a T-shirt. You can buy the T-shirt with the brain maps on. So let me tell you about brain size, for example. So if the x-axis is age, and when you're 20, your brain is at its largest, there's a steady loss of brain tissue till you're about 60, and then a more rapid loss of brain tissue into old age. But look at how different people are. So this is just a chart of brain size measures across 18,000 people from 35 countries. And you can see that among the 20-year-olds, I mean, one person's brain might be double the size of another. So this is a very complicated situation. This is the ENIGMA lifespan data viewer, where you can norm your patient's data versus the aggregate. If a patient has an MRI scan, you can compare their metrics with everyone else and maybe a matched population that makes sense for them. And you can say, well, for various brain regions, the hippocampus, the thalamus and amygdala, what are the trajectories? This is just the size, the volume of parts of the brain, but certainly these diseases affect that as well. And then you can establish a normal range. And this system is actually called Centile Brain. It's online. You can upload measures from your patients and see. In this case, this is a chart for women, the volume of their left hippocampus. And it's just a percentile chart. So there'd be like the average volume for women of a certain age, and then the different percentiles. And you could not only see where a patient lies within this range, but also if they're treated or if they have a certain intervention, do they rise in the ranks, so to speak? Do their percentiles get a little better, or just what's going on? So you could say, okay, that's normal variation. How do diseases, and for that matter, genetic variants, affect these brain scans? And this may be interesting. Brain measures are certainly a good target for genetic analysis. It may be easier to find genetic variants that affect the brain, that affect disease. But a lot of the time, this has required nearly 100,000 people to understand what it is in the DNA that's different between a patient group and controls. So here's a skeptic. Nick Martin was the president of the Behavioral Genetics Association. He said, if you want to discover genes that affect the brain, you'll need brain scans from 50,000 people, just because images are more expensive to collect than measures of height or weight. It doesn't change the power calculations. And he, to a certain extent, was right. So when ENIGMA began and was screening DNA for markers that affect brain regional volumes, it took about 30,000 people to discover them. And the reason is, it's not that the effects are negligible, it's that you're testing over 1 million possibilities. As you go across the genome, I mean, you don't want to have an accidental discovery that's never replicated later on. So by gradual replication and meta-analysis, these markers emerged as being important for influencing, in this case, the volume of the brain. How does it work? You collect a saliva sample, you sequence the patient, you measure at every point in the genome whether the variation there influences something about the brain. This could be the size of the brain, but it could be the level of amyloid. It could be the function of the brain in children. This is just an example. And just to give you an analogy, if we wanted to find genetic variants that are associated with a disease like Alzheimer's, the x-axis is your genome, the y-axis is the strength of evidence of association. There are about 80 markers in the genome that are associated with Alzheimer's disease, the most common being the ApoE, apolipoprotein E gene. There's variants there that affect your Alzheimer risk by a factor of 3 or a factor of 15 if you carry 2 copies of that gene. And also in other diseases, you can look at schizophrenia. Are there any genetic differences between schizophrenia patients and controls? And on average, there are differences in the immune system, the major histocompatibility complex. And as you add patients to these, more and more hotspots in the genome come out that have an association with a disorder. And the goal is to treat them. I mean, you would treat the protein pathways that are influenced by these directly. So ENIGMA uses this model. We all compute brain measures from our brain scans. They can be structural scans on the left or diffusion images on the right that look at microstructure. You can test associations between these brain measures and all the 1 million markers in the genome that might affect them. And then you combine the information across the world, a meta-analysis. So if you have 10 patients, that's fine. If you have 10,000 patients, that's fine. Just the evidence would be factored in from the sample size. And this was hugely interesting and successful. So 6 genetic markers or loci in the genome were discovered where all of us vary. We have differences in our genetic code. And those differences in our genetic code affect the size of the hippocampus, the brain's major memory system. And so, in fact, that's a model of the hippocampus there from MRI. There were markers that seem to have to do with cell death, apoptosis, that influence just, in this case, the volume of the hippocampus. A colleague of ours who's working actually in Dagestan said the location in the genome that you found affects the hippocampus. In my patients, and they're shown here, they're in an ethnic isolate in the Caucasus Mountains of Dagestan, we found that area of the genome is influencing their risk for mental retardation, their risk for, in fact, psychosis. That same marker in the genome that's affecting the mental health of her patients, these are her patients here in the Caucasus Highlands, she found, Kazima Bulayeva is her name, she found that this locus not only erodes hippocampal volume in everybody, but when it's mutated, influences psychosis risk in her patients. So this carried on. Novel genetic loci were associated with hippocampal volume. You can ask questions. You can say, well, Paul, you told me about this Alzheimer's gene, this gene that a quarter of us carry, this ApoE4 risk that triples your risk for Alzheimer's. Is that eroding hippocampal volume? And it is. You can go look in the brain scans. You can look at the people that carry the Alzheimer's risk gene, and you can see that by the time they are in their 70s, their hippocampal volume is shifted by about 1 standard deviation away from everyone else, and by their 80s, 2 standard deviations from everyone else. So this brain scanning project is providing essentially mechanisms that might underlie the mode of action for these disease risk genes in the brain. The other thing is that the genes that affect different parts of the brain are different. So one of the genes that affects the size of the brain, the volume of the brain, MAPT, it controls tau, which is a very important molecule in your brain, tau protein. It's a major risk gene for Parkinson's disease. And so this is, let's just show you it, a genetic screen for markers that influence your risk for Parkinson's disease. There's MAPT, the tau gene, over on chromosome 17 on the right. There's another important gene, synuclein alpha, that affects dopamine release in synaptic vesicles, and it makes a lot of sense for Parkinson's disease. So what's emerging is a picture where some of the disease risk genes are also genes that affect important parts of the brain. I mean, this was actually just the size of the brain. So you can begin to see a link or a bridge being drawn between disease genetics, this DNA marker affects my risk for disease, and brain genetics, where you say this DNA marker is affecting hippocampal volume or a certain genetic program that we can identify in the scans. So this kept going, more and more loci affecting the brain, and now there's over 500 of them that affect the volumes of these things. Now, what practically can you do with this? Well, it's sort of interesting. If this many markers in our DNA affect the brain, you can actually predict brain measures from them. Not completely, I mean, not everything about our brain is genetic, but about 9% of the variability in brain structure can be explained just from a saliva test. You can spit in a vial and have your genome sequenced, and even these disorders of different brain structures, a lot of the variation is dependent on markers that we've now discovered in the DNA. Now, drug companies are interested in this as well. So you could say, Paul, did any of these people come back? Did they have 2 brain scans? Because if they had 2 brain scans, we can see how quickly they're losing brain tissue. And then if there's any markers in the genome that are causing fast brain tissue loss, we'll be able to find them, find drugs that might modulate them or interfere with the cascade. So this ENIGMA project, 10,000 people were scanned twice, about 2 years apart. And Rachel Brower and Hilke Hulshoff-Paul asked this question. They said, is there anything in the DNA, our DNA genome of 3 billion base pairs, is there anything that predicts the rate at which these people are losing brain tissue? And there is. This is in Nature Neuroscience last year. 15 loci in the genome where if you carry a variant at these loci, your brain tissue losses faster. Now, this is early days, but it's the beginning of a roadmap of brain degenerative loci that we can target with treatments. Now you say, Paul, I like genetics, but I'm more interested in patients. I want to see if any of the patients with these conditions have differences in brain scans that we can understand. So ENIGMA published the largest neuroimaging studies of schizophrenia, bipolar, major depression, PTSD, substance use, which goes from, as you know, meth, alcohol, cocaine, cannabis, opiates, and then these developmental conditions, autism, ADHD, OCD. And then moving back to neurology, the very large-scale studies of epilepsy, Parkinson's, ataxia, and TBI. So just to say how these are done, here's a schizophrenia study that is going on at 33 sites, and this is where they're collecting the scans of the patients. And they, just like we said, measure all the parts of the brain, the size of the brain, the activity, the microstructure, and they compare notes. So those are 33 cohorts. 25 of them found that in schizophrenia, there's a thinning of the fusiform gyrus. It's a very important part of the brain involved in face recognition. And the patients had about half a standard deviation shift compared to healthy people for that measure. And you can see the strength in numbers. So if 25 of these cohorts discovered that this was a brain abnormality in schizophrenia, it pretty sure is true. And the cohorts that didn't typically had smaller samples. So a lot of these studies are fairly small. And you can make a map of the brain and say, where does schizophrenia affect the brain? Well, schizophrenia affects the brain everywhere. The only region not affected is the visual cortex, half a standard deviation difference from controls. And you can look at other diseases. So here's a map of the brain structural differences in major depression, in bipolar disorder. I'll show you a summary at the end. We'll just look at each disorder. A deletion syndrome that affects children, epilepsy. These are very, very large-scale studies of thousands and thousands of patients. And the volume, the gray matter thickness in this case of the brain, is compared between the patients and the controls. And as you look at these different disorders, you'll notice some interesting things. In autism, for example, the thickness of the frontal cortex is bigger. It's very perplexing why that should be. There might be an overgrowth or different growth in autism in childhood. But then as you look at some of the other disorders, they are more in line with expectations. People that are addicted to alcohol, there's huge frontal atrophy, there's cingulate atrophy, there's patterns for each of these. So as you look at the picture here, these are the structural imaging findings, and there are major differences. Major depression up there in the middle at the top, only the superior temporal gyrus, and in fact the cingulate are showing differences. But as you go on the top right to bipolar disorder, much more of the brain is affected. And it's beginning to give a brain cartography for some of these multiple disorders. And you can use this for differential diagnosis. So in depression, as we said, the cingulate up there on the top right, the limbic system is affected. In bipolar disorder, this is a study of 6,000 people who are scanned. The frontal areas, in bipolar, one of the issues is behavioral inhibition. The frontal areas are showing abnormalities, so it makes sense. And as you look at, for example, schizophrenia, bipolar, depression, ADHD, you could imagine a sort of barcode where aggregating all this data from 33 countries, there are certain brain regions that are telltale signs of one condition, maybe the mood disorders, for example. And ADHD, it's been very challenging to find a structural imaging biomarker of ADHD. But in this study, the basal ganglia, which also makes sense in ADHD, are showing abnormalities more so than any other part of the brain, at least structurally. Now, the people working in other countries are doing a great job of replicating and extending these findings. So this is Ryota Hashimoto, who leads ENIGMA's efforts in Japan, Kokoro's study in Japan. And he's scanning people all over Japan. You'll see in Western Japan, this very famous area where people age very well. Their longevity is very, very high in Western Japan. And isn't it interesting to look at the scans and see if there's anything about their culture, their genetics, their diet, or anything else? We could learn a lot about this. And you can do these with all kinds of brain mapping methods. I talked about structure. You can do it with function. You can do it with any other type of brain scan. And as the studies get richer and with higher numbers of individuals in them, the patterns associated with the disorders, in this case obsessive compulsive disorder, they begin to settle down and coalesce and implicate regions that are consistently associated with the different conditions. So this is ENIGMA OCD. It's the part of ENIGMA that deals with obsessive compulsive disorder. And they've said, you know, we're starting to puzzle this out. The brain differences in OCD, they seem to be in the thalamus, the sagittal stratum. But the reason we haven't found these consistently in the past is that the heterogeneity is enormous. I mean, obviously, a child who's had OCD for a long time might have more profound brain differences from someone who's recently diagnosed. The duration of illness matters. The types of medication matter. So you can imagine that to first order, having a brain map of a disorder is possibly useful. But much more interesting is what is it about the patients and how they're treated and their life experiences that make them do a little better than others, or the opposite. Groups looking at eating disorders, this is a very good story. They found that even though anorexia has the greatest effect on the brain of any psychiatric condition, the brain volumes are one standard deviation off from healthy folks, this is the one disorder that has been found to be very, very treatable. And so people with brain imaging, after they've been treated for their anorexia, they show restoration. They show restoration in these brain metrics that previously were thought to be, to some degree, irreparable because they're structural markers. Lots of age-old questions. So for 50 years, there's been an assertion that in schizophrenia, the hemispheric asymmetry, the laterality of the brain, is different. And this is just coming out in PNAS. Dick Shiven looked across the world in all the schizophrenia data that could be collected, 46 data sets from 5,000 schizophrenia patients, and he found that that is true. The laterality of the brain is reduced in schizophrenia. And this was a hypothesis by Eve Johnston and Tim Crow in England in the 70s that there's been a whole lot of arguing about for a long time, but now there's finally some definitive data. Some diseases are progressive, so it doesn't really make sense to make an average picture of them. So for disorders such as Parkinson's, where there are many stages, we have the patient data broken up into stages. Stage 1, 2, 3, 4, 5 in the Hone and Yahr staging system are used for Parkinson's. And you can begin to see patterns as the pathology progresses in the brain, maybe modulators, how these patients are treated, maybe with L-DOPA, maybe with deep brain stimulation. You can use these as references to see if they're doing the same as the other patients. And you can use it for staging. You can say, well, look, these are the basal ganglia. We've sort of broken them up here, the hippocampus, the caudate, the putamen, we've broken them up. The atrophy that happens in Parkinson's at different stages occurs in a certain sequence. So you can use these brain scans as a reference to see whether a patient's at a certain biological stage. Some of the brain diseases don't affect the cerebrum, or not as much. So in ataxia, most of their efforts in the ENIGMA group on ataxia, pooling data from 10 sites, is to map the cerebellum. I mean, the cerebellum is the target. It's the main area involved in motor disorders, certainly the ataxias. So they've been producing some great findings that track the trajectory of ataxias in the cerebellum. It's not often thought of as a progressive condition, but relate those to early onset, later onset, and different types of treatment. And a lot of the tools they develop can be used by other groups. So ataxia, they've developed a lot of tools for the cerebellum. The other groups studying other disorders can say, well, hey, maybe we should look at the cerebellum. There's a lot of differences that potentially are found in other disorders as well. You can also make predictive models. So if you scan a patient today or a family member today who's worried about their risk for a condition, you can see if there's anything in the brain scans. There's been sessions at this meeting on AI and machine learning. If there's anything in the brain scans that foreshadows future development disease or resilience. And so this is a very, very interesting area that researchers are looking at. Okay, last 5 minutes. What are the new directions? What are the future directions? Well, one of them that many of you are very active in is AI and deep learning. There's a lot of interest in looking at brain connectivity, functional connectivity of the brain. And then finally, predicting treatment effects or selecting treatments that are best. There's new ENIGMA groups on neuromodulation that look at TMS, which is often used in depression and OCD, and neurofeedback. And so neurofeedback is sort of an interesting area where there's no explicit intervention, but the patients are asked to modify their behavior or thinking in certain ways, and we're trying to meta-analyze what those effects are when neurofeedback is used in different conditions. Now, just to tell you a little bit about AI, you can certainly, and this is work we did with Chao Ganyan and Bin Lu in Beijing, you can certainly feed in a brain scan and ask an AI method to tell you what disease the patients have. And just to be honest about how these methods work, the accuracy in detecting a disease like Alzheimer's disease, which actually has a very strong signature in brain scans, the accuracy of the best method is about 91%. There was a presentation yesterday showing around 90% as well. The more training data, the better. And so these AI methods are very interested in seeing scans from as many patients as possible. They are based in computer vision. So these AI methods that are used to recognize cars, flags, cups, other objects, same thing will work with 3D MRIs. They're modified a little bit, but it's very interesting. So there's huge activity in ENIGMA and many of the groups working here in adapting AI computer vision methods to work with brain scans and to see which methods are best at diagnosing and prognosing different diseases. Second area is connectomics. So this type of brain scan is used preoperatively to map the pathways in the brain. It's a diffusion MRI scan here mapped with tractography. And you can look at the details of brain connectivity, the fiber pathways. This is a section, a sagittal section that goes through the limbic system. Many of you will recognize the cingulate that looks like a big freeway there in the middle. But you can use all of these as inputs to the genetic and disease analyses that I've talked about. You can say, what is the roadmap of connectivity of the brain? How are the connections doing? I mean, can we represent that as a matrix? And then can we screen the genome? What is the genetics of the connectome? Can we search the connections of the brain and see if there are any markers in the genome that guide its development or its deterioration? You can look at tractometry across diseases. So in ENIGMA, we're pulling out the cingulum, the corpus callosum, many of these different neural pathways, and asking, what is the disease doing and what are the treatments doing to these individual pathways? And then let's not forget the environment. So I talked a lot about genetics. There's certain things in the environment, I mean, either toxins, pollutants, urban density, lead, radon, many things that affect the brain and brain health. And then also social determinants of health. You can look in the environment and see whether folks have access to healthcare, what is their socioeconomic condition. And so these are being measured as well in a very exciting new group called ENIGMA Environment, where brain scans are geocoded. You link where the person was scanned to their address, and then through their address you can go and look at the, in this case, pollution. And so, I mean, in Los Angeles the pollution isn't that great, but there's many parts of the world with bad pollution. And it's interesting to break it up. I mean, ammonium, nitrate, sulfate, I mean, you could link any number of these things to the brain scans and see if you can pick up any association. One last thing is a cultural thing. So we've learned that global participation is important for science. Now, there's no better protagonist than Dr. Kartab here in developing such a wonderful society that spans the entire world, doing international brain mapping. It's politically beneficial, not just data. It's not that we all are good at collecting data. It's politically beneficial when projects are led from different countries. So you'll hear a lot of computer scientists saying, well, I mean, it's just the information. But I mean, the expertise and the cultural perspectives, I mean, all of you know this from your work in many countries of the world, are often what really makes the difference in answering some of these questions. So we have an India ENIGMA initiative. I was actually there last week. We're collecting brain scans in India. And a lot of the questions are the things going on in India that protect our brain or are harmful for the brain. Certainly the social support of elderly folks in India is better. The diet's different. The many public health things are different. So it's very, very interesting to look at that. ENIGMA has a very active group in Thailand. They're interested, this group in particular is interested at the Thai Red Cross in infectious disease. And so they have an ENIGMA project to compare brains of children that have maternal HIV infection with other children from around the world. We talked about Japanese ENIGMA, their efforts to compare the brain differences in Japan with here. There's certain parts of the world that are not traditionally working with people in the US. Very, very active groups in these parts of the world. In Qatar, there's an ENIGMA initiative. They have a very strong genetics biobank in Qatar. It's a very, very ethnically homogeneous population where they've sequenced many, many people. And so just to wrap up, this is what we're doing this year. We're analyzing diffusion MRI and brain connectivity across the world and across disorders. There's resting state fMRI that's piloting in 15 disorders. There's some work on machine learning and deep learning. Why would you do that? Well, you would want to do patient clustering and subtyping, certainly predicting treatment response or best treatments for given types of patients from their biomarkers and certainly from their neuropsych. And then also genomics. I mean, do the different subtypes or dimensions of disease have different genomic drivers? For maybe some diseases, maybe they do. Maybe other disorders, it's not genetic. And I think a lot of the partnerships, many of you making connections this week, people in Japan, India, Pakistan, Maheen Adamson and ENIGMA spearheading an effort in Pakistan, that will in the end help disentangle the social and cultural and environmental effects on brain health. It's not all genetic by any means. And so this effort to do geocoding by ENIGMA environment is a bridge between the brain scans and all the ambient data that people are collecting on health-related environmental measures. So do follow us on YouTube. You're very, very welcome to go look at ENIGMA's website. And if you're collecting brain scans from patients, you're very, very welcome to join us. You can look up ENIGMA. And big thank you to Babak for the invitation. Big thank you to all of you for coming on a Sunday morning and being here with us today. And it's been a real pleasure to do this. And thanks. Some of the people pictured here are collecting the data for the projects I've shown today. So thanks very much.
Years ago at a neuroscience meeting, an extremely impressive poster. This is by an order of magnitude the most impressive presentation I've ever seen. So congratulations on your really excellent work. I did not see you focus very much on the brainstem, which we've been tremendously interested in, done tractography on the brainstem. But that's not what I want you to comment on, because I'm sure we'll get around to that. I want to ask you some political questions. I didn't see any mention of the Alzheimer's disease neuroimaging initiative, which is also at USC. And in the VA, we're having a lot of trouble coordinating these sort of things. And if you can coordinate all the people in the world or all the countries in the world like this, could you help us with the VA and get the VA organized? And how about all these homeless people we have on the street we need to brain scan and get them into something else? How can you help us more?
Yeah, thanks, Wes. It's very generous of you, and I really appreciate what you said. I think for Alzheimer's, and you know this, I mean, Wes is a leader in the field of Alzheimer's. It's been a little different, and the reason is NIH has funded massive public databases, particularly of neuroimaging, but also of clinical and genomic data. And a lot of the data is public, and so you can get ADNI data from the internet and analyze it. So there was not quite, at least initially, the same impetus to get groups together. If you were looking for brain scans of people with psychiatric conditions, it would be much, much more difficult than in Alzheimer's. So, but I think your point's a very important one. All of the efforts that maybe you and I and others here could come up with to bring people in the field together would be immensely valuable, particularly now that there are treatments, or at least some treatments with some mechanisms that are very complex. And you might want to have, as you know, brain scans and neuropsych compared across the world from people taking those things.
I want to say one other thing. Not terribly interested in treatment. I don't want my Alzheimer's disease treated. I want it prevented.
Yeah.
And I don't want to have schizophrenia. I want schizophrenia prevented. And I think the sort of thing you're doing, maybe could lead to that in that direction. We're going to have a session in here in 10 minutes on our Alzheimer's sessions. At 3:30 today, we're going to have a session. I don't know if there's going to be anybody left, but anybody who's left, we're going to have a session. I'm going to give an introduction to why I think we should move forward with prevention. And anybody who wants to stay, we're going to try and develop a paper, and maybe your name could be on the paper. So the enticement to stay until 3:30.
Thanks, Wes. Thanks so much. David.
Paul, as always, brilliant. I always learn something, even if I've seen him for the fourth time. I wonder if you could comment on something I find very provocative, in that the eating disorders, we see restoration. Those of us who are graybeards were taught, well, the brain doesn't, once it's damaged, it doesn't ever really heal, and now we know that's not true. But what's your speculation? Could it be something like modulation in the inflammasome, that something is actually healing, and then there's an indirect effect on structure and function? Because that's one of the things I found. I get the joy of seeing this data when it's hot off the presses at our scientific meetings, but I'm still not clear what might be going on there.
Thanks, David. I think that's a great question. So the recovery of brain structure, which is really prominent in anorexia, I mean, if you believe the old-fashioned theories of neuron loss and neurons being irreplaceable, even if some of it is neurogenesis, it may not be a lot. So could it be, and I think your hypothesis is a good one, maybe there's an inflammatory reaction, maybe there's myelin coming back and being restored, maybe there's a glial restorative response. Having said that, a lot of these folks in that study are adolescents, and so there might be a lot of things going on for them that are positive and can help them get back on track. But it's a great question. Maybe we can look at this with, as you know, other methods like spectroscopy that can get into the depths of what is going on with the cells. Great question.
Paul, great work and mind-blowing. You know, you think about, we can identify a schizophrenic before their break at 22. My passion's like in corrections, brain injury and corrections. And the precision that you're talking about, irritability, sleep, domestic violence, are we also going into corrections? That's so interesting that these brain scans of somebody that has domestic violence, maybe both sides, the perpetrator and the victim. Who's doing that work?
Yeah, so the domestic violence work is led by Kari Esopenko at Rutgers, and there's a related group led by Mark Shiroshi here at USC on child abuse. Now, you could say, well, how on earth would you do a study of these things? That must be very, very difficult to do. I mean, would the person really be able to get involved in research? So she has a fantastic support network. It's called ENIGMA IPV, and it's a care network for people that are, they're in an environment where there's domestic violence. It's not always easy to reach out to someone else. So Kari Esopenko's her name. She's leading the ENIGMA IPV group. Obviously there, the goals are a little different. It's not just to show that there are brain differences in someone with brain trauma, but to try and understand, how to get them better help and how to get them to recover and what types of things work there. So that's really, really important area, very exciting area.
Yeah. I just want to think, the retina scannings have just taken off. So we've got a piece of the brain that moves, or is there ideas of putting a retinal scan group that we can go ahead and connect this? It's even more accessible than the MRIs.
Yeah, I really like your idea. So, I mean, many of you that go and have an eye test will have OCT, or, the ophthalmologist will have all sorts of even deeper methods looking at particularly vascular changes in the eye that often co-occur with the things going on in the brain. So I think that's a wonderful idea. I mean, if it makes sense to have multiple groups come together and analyze their data together, this type of setup would likely be very valuable, and I'm sure maybe there are studies in retinal.
Well, and the new instruments now do OCT, FA, and so the number of scans I do per day.
Oh, would be fantastic.
Somebody needs to get this data and use it. Thank you.
Sure.
Good morning. First of all, that talk was incredible. I'm so happy I woke up this morning and came. So. I'm Abhi Haridas. I'm a neurosurgeon from Tampa. I'm an adult and pediatric neurosurgeon. So my question is, looking at your talk, there's always a disconnect between the surgical community and the imaging community.
Yeah.
And as a surgeon, when we intervene, we sometimes end up in, we have to have a prognosis to tell a parent, especially in the posterior fossa tumors for children. We are constantly in that area. And does your organization have a bridge for us to share that data? And I want to take it one step further. As a pediatric neurosurgeon, I see also a lot of pregnant women with fetal MRIs. That's an enigma. I have no idea what to do with that information. So it's almost like I want to lateral pass it, but I also need to understand, is that something that you've had trouble with too? And I'm hoping we can collaborate on that too at some point.
You know, I really appreciate your comment. I mean, certainly post-surgical recovery is an area where we could do a lot together. I mean, we have in ENIGMA groups on brain trauma, concussion, things like this. In general, the outlook is good, as you know. I mean, from your own work, the outlook is generally good. But having imaging is tremendously reassuring. And so having a scan that can say, look, this looks like you're doing well, it's exactly what we would expect, the course of this is going well. Having prognostic tests, particularly after surgery, as you know, can be tremendously useful. And that's a time period where folks, as you know, have more time. There's not so much urgency to do these things. So being in a research study that is longitudinal is something that's interesting to them. And so that may be an area that we could certainly work together on post-surgical recovery. The fetal MRI is a very exciting area. So it's very new. The methods for volumetric quantification are getting to be really, really good. And as you know, in utero diagnosis is a really, really important area. So if there were a group that were interested in fetal MRI, I'm sure we could nurture and support them to get something going on prognostics.
Thank you very much.
Sure. Thank you.
Excellent work. Remarkable work. Congratulations, Paul. I think, come here, don't go. I think there are great areas of collaboration. We have 100,000 scientists on our network globally. And Paul and I talk about potential joint conventions, so we could. But what you have created is scientifically marvelous. We have to turn it to policy. So that's what we did. We started the BRAIN Initiative with President Obama. Under President Biden, we are starting a biotech park for brain in LA. So it's called Brain Technology Innovation Park. So it's really taking this concept and fast track it to therapeutics. And as Dr. Abhilash said, our group is a much more interdisciplinary, clinically relevant intraoperative mapping and so on. So we could bring that to the table. So with that, I congratulate you for your Pioneer in Medicine Award.
Appreciate
it was
it
great.
very much.
Round of applause for him. And we're also appointing him on our board of directors. So another
Very kind. Very, very kind.
pleasure. Thank you very much, everybody. Thank you, Paul. Thank you.
Thanks so much.
We're going to go to the sessions. Okay, thank you.